Welcome to Rad Onc Smart Review, RadBio edition.
Great to be here.
Today we're going to take a deep dive into tumor assays in radiation biology.
Right.
So moving beyond the petri dish, really, we're heading into the complexity of living systems.
Exactly. Seeing how tumors actually respond to radiation inside an organism.
We'll be looking at the key experiments, right? The assays used to measure tumor response.
Yeah, things like tumor control, cell survival within the tumor, and how growth gets perturbed.
It's like getting the real story before treatments even hit the clinic.
That's a good way to put it.
Okay, so just to kind of set the stage, we've talked before about how radiation hits individual cells.
Mm-hmm.
The survival curves, the linear quadratic model, mostly from in vitro work, right? Lab-based stuff.
Exactly, yeah. We covered how radiation damages DNA, stops cells dividing, target theory, LQ model, they help relate dose to survival.
Right, for single cells in isolation. But here's where it gets really interesting, I think. How does this all work in a real tumor? Inside a living animal, in vivo?
Big difference.
Because a tumor isn't just like loose cells floating around.
Not at all. It's a whole ecosystem, isn't it? Got its own blood supply, tissues, immune cells, signals flying everywhere.
Plus the animal itself, the host.
Right, the host factors. So while those in vitro clonogenic assays are, super useful for basics, we really need different tools to see if a treatment works in that complex living setup.
Makes sense. Okay, let's dive into the first area then. Assays that measure if we can actually, cure the tumor or control it.
Right, the big outcome.
And the gold standard here seems to be the TCD50 assay. Tumor control dose 50%.
Spot on. TCD50. That's really the benchmark in preclinical work for figuring out the dose needed to cure a tumor.
Okay, TCD50. What's it actually telling us?
So the definition is it's the radiation dose needed for permanent local control.
Permanent local control.
In 50% of the tumors you treat. And permanent local control means the tumor completely vanishes and doesn't come back during the observation time.
Completely gone, no recurrence.
Yeah.
Got it. So how do researchers actually figure out this TCD50 number? What's the setup?
Okay, imagine you've got groups of identical mice, same genetics.
Syngeneic.
Yeah, exactly. And they all have the same type of tumor grown to about the same size.
Okay.
You divide them into groups. Each group gets a different single radiation dose aimed right at the tumor.
So maybe one group gets 40 grays, another 45, another 50. Something like that.
Precisely. A range of doses.
Then what? Just watch them.
Pretty much. You observe them carefully for a long time, maybe 120 days, could be longer.
Okay.
And for each dose group, you count how many tumors were totally wiped out and stayed gone.
Ah, so you get a percentage of controlled tumors for each dose.
You got it. Dose versus percent controlled.
And then you plot this data.
Yep. You plot percent controlled against the radiation dose. And what you usually see is an S-shaped curve, a sigmoid curve.
Okay, that classic dose-response shape.
Right. And the TCD50 is simply the dose on that curve that lines up with 50% control.
So you just read it off the graph, essentially.
Basically, yeah. It visually shows how the probability of cure increases with dose. It's a really key concept for figuring out treatment regimens.
Makes sense. And you can do this for different treatment schedules, too, like not just single doses.
Absolutely. You can generate TCD50 curves for single doses or 2 fractions, 10 fractions, whatever schedule you're testing.
And what do those curves look like compared to each other?
Well, what's interesting is if you compare, say, a single-dose curve to a 10-fraction curve, the 10-fraction curve will usually be shifted to the right.
Shifted right, meaning you need a higher dose.
A higher total dose, yeah, to get the same level of control, like 50%.
Why that?
It's mostly because giving the dose in smaller chunks, fractionated, allows the tumor cells time to repair some of that radiation damage between doses.
Ah, okay. That fits right in with the R's of radiobiology. Repair, specifically. So why is knowing this TCD50 value so critical? What's the big takeaway?
Well, the TCD50 is thought to tell you the dose needed to kill all the clonogenic cells in at least half the tumor.
All the cells capable of regrowing the tumor.
Right, including those really tough ones, the cancer stem cells or tumor-initiating cells. They're often more resistant.
Oh, okay.
So if you achieve TCD50, it suggests you've probably managed to sterilize even those critical resistant cells in a good chunk of the tumors. It really speaks to overcoming that resistance barrier.
That's a really important point. Okay, so TCD50 is about the cure, the ultimate outcome. What if we want to look closer at how many individual cells survive inside the tumor after radiation?
Good question. That takes us to a different set of assays, ones that measure cell survival in vivo.
Like the in vitro clonogenic assays, but inside the animal.
Pretty much, yeah. Trying to quantify survival, but accounting for that whole complex tumor microenvironment.
Okay, so not counting colonies in a dish. How do you count survivors in a living tumor?
One key way is the dilution assay. You might also hear it called the TD50 assay, tumor dose 50%.
TD50. Okay, what's the idea there?
The TD50 is defined as the number of viable tumor cells you need to inject into a recipient animal to get a tumor to grow to take in 50% of those animals.
So it's about how many cells it takes to start a new tumor.
Exactly. Their tumorigenic potential.
How's the experiment run?
Okay, first, you take tumor cells from control animals. No radiation.
Untreated tumors.
Right. You make a cell suspension and prepare serial dilutions. So you might have tubes estimated to contain, say, 8 cells per shot, 4 cells, 2 cells, 1 cell.
A range of low cell numbers.
Yeah. Then you inject each dilution into groups of recipient animals. Again, usually syngeneic, genetically identical mice.
Why identical mice again?
It's crucial. You want to make sure any tumor that grows is from the injected cells, not because the mouse's immune system is reacting to something foreign.
Got it. So you inject these different cell numbers.
Then you watch, see which groups develop tumors. From that, you figure out the control TD50, the number of cells it took to get tumors in half the animals. Let's say, just for example, it's 3 cells.
Okay, 3 untreated cells equals 50% tumor take. Now, how do you use this to see radiation effects?
Now you do the whole thing again, but you start with cells from animals whose tumors were irradiated in vivo, say with 10 grays.
Right.
Now, because radiation killed a lot of cells, you'll need to inject more cells to get tumors. So maybe your dilutions are now 64, 32, 16, 8 cells per injection.
Higher numbers needed.
Definitely. You run the experiment, watch for tumors, and find the TD50 for this irradiated group. Maybe this time it takes 32 cells to get 50% tumor take.
Okay, so control TD50 was 3, irradiated TD50 is 32. How do we get surviving fraction from that?
Simple calculation. Surviving fraction, SF, is just the control TD50 divided by the irradiated TD50.
Uh-oh.
So in our example, 3 divided by 32, that equals 0.094.
So about 9.4% survival after 10 grays.
Exactly. And you absolutely need to remember that calculation-controlled TD50 over irradiated TD50 equals surviving fraction.
Is this like a classic board exam question?
Oh, yeah, definitely. The TD50 concept and the SF calculation, very common. And what's cool is by doing this at different radiation doses, you can actually build in vivo survival curves.
Really? Like the ones we see from in vitro work?
Yeah, and these in vivo curves were super important. They helped confirm things like the oxygen effect, showing that hypoxic cells in a tumor really do need much higher doses to be killed. That relates directly to the OER, the oxygen enhancement ratio.
Wow, okay. So these in vivo assays confirmed fundamental radiobiology.
Absolutely, and that has real clinical implications for thinking about things like treating hypoxic tumors.
Fascinating. Okay, you mentioned another survival assay too, the lung tumor colony assay.
Yes, the lung colony assay. Dick Hill did a lot of pioneering work on this. It looks at the ability of tumor cells treated in vivo to then go and form colonies in the lungs of recipient animals.
So it's about survival and metastatic potential.
Kind of, yeah. It tells you about cells that survive treatment and retain the ability to lodge somewhere else and grow.
How does that one work?
Okay, you grow a tumor in a donor animal, usually under the skin. Then you treat it, irradiate it, for example.
Treat it in situ.
Right. Then, after treatment, you surgically remove the tumor, process it into a single-cell suspension.
Like before.
Yep. But this time, you inject a known number of these cells intravenously, often into the tail vein of recipient mice.
5E injection. So the cells go everywhere.
They circulate through the bloodstream, yeah. And the idea is any surviving clonogenic cells can get trapped in the lung capillaries and start growing.
Forming little tumor colonies in the lungs.
Exactly. So after a set time, maybe 18 to 21 days, you examine the lungs of the recipient mice and count the visible tumor colonies on the surface.
And the number of colonies tells you.
It's directly proportional to the number of surviving clonogenic cells that were in that initial injection.
So it's another way to compare treatment effectiveness on cell survival.
Right, and it gives you that extra layer about metastatic capability, which is also highly relevant for board review.
Okay, great. Now there was one more survival assay you mentioned, a hybrid. The in vivo, in vitro assay.
Ah, yes. This one tries to get the best of both worlds. You treat the tumor in the living animal, but then you measure survival using precise in vitro methods.
Okay, how does that work, combining the two?
So you grow the tumor in the host animal, irradiate it in vivo.
Right.
Then very soon after irradiation, you excise the tumor, take it to the lab. Then you process it, mince it, use enzymes like trypsin to get a single-cell suspension, just like you would for a normal in vitro assay.
Okay.
You count the cells, plate known numbers onto petri dishes, incubate them, and then, stain and count the colonies that grow.
So the final measurement is just like a standard lab clonogenic assay, calculating plating efficiency and surviving fraction.
Exactly the same calculation. But the key advantage is the cells received their radiation dose under those realistic in vivo conditions.
With hypoxia, cell contacts, all that stuff.
Precisely. So you get the relevance of in vivo treatment combined with the quantitative precision of in vitro colony counting. Another really important one to know.
Got it. Okay, so we've covered assays for cure TCD50 and assays for cell survival TD50, lone colony, in vivo, in vitro. What about just looking at how the tumor grows after treatment?
Right. That brings us to assays measuring growth perturbation, how treatment messes with the tumor's growth pattern over time.
And the main one here is the tumor growth delay assay.
That's the big one, yes. Tumor growth delay. It's widely seen as the gold standard preclinical assay for seeing how well therapies slow down tumor growth.
Gold standard. Why is that?
Well, importantly, results from this assay often correlate pretty well with how treatments perform in actual human clinical trials, especially with achieving clinical remission.
Ah, okay. That's a strong link.
Yeah.
So how do we define and measure this growth delay?
Okay, basically, it's the extra time it takes for a treated tumor to grow to a certain size compared to an untreated control tumor.
Extra time. Makes sense. How's it calculated?
There are two main ways. One way, less common maybe, is just measuring how long it takes an irradiated tumor to grow back to the size it was at the time of irradiation.
Okay, time to regrow to original size.
But the more standard way is this. You define an initial size, size A, when treatment starts, and a target larger size, size B.
Right.
You measure the time it takes the control tumors to go from A to B. Let's call that T control.
Okay.
And you measure the time it takes the treated tumors to go from A to B. Call that T treated. The growth delay is just the difference. T treated minus T control. That extra time the treatment bought you, remember that. Growth delay, T treated, T control to reach size B.
Got it. T treated minus T control. So walk me through the experiment itself.
Okay. Implant tumor cells in animals. Let them grow to size A.
Start point.
Right. Randomly split them into groups. One gets treated, say, irradiated. One is the control group.
Untreated.
Yep. Then you regularly measure the tumors in both groups, usually with calipers, calculate the volume.
Keep tracking growth.
Keep tracking until the tumors in both groups reach that target size B. Then you look at your growth curves, volume versus time for each group. From the curves, you find the average time each group took to get from A to B. Calculate the difference. That's your growth delay.
Okay. And what's the relationship between radiation dose and growth delay, typically?
Generally, higher doses give you longer growth delays. Makes sense, right? More cell kill takes longer to regrow.
Yeah.
You often see plots showing growth delay increasing as the radiation dose increases. Studies might compare X-rays and neutrons, for example, and show neutrons give more growth delay per gray for certain tumors, like that rat rhabdomyosarcoma model.
Interesting. And you said this correlates well with clinical results.
Yeah, there's pretty good evidence for that. Studies have compared growth delay caused by chemo drugs in human tumor xenografts.
Human tumors in mice.
Right. They compare that delay to the clinical complete remission rates for those same drugs in patients with similar tumors, and often there's a decent correlation. So a longer growth delay in the mouse model often predicts a higher chance of complete remission in humans. It really supports using growth delay as a key preclinical endpoint.
That's powerful. Definitely highlights its clinical relevance. Okay, so growth delay is key. You also mentioned briefly a tumor regression assay. How's that different?
Regression is simpler. It just measures how much the tumor shrinks after treatment.
Just shrinkage.
Yeah. You measure the size before, measure it after, see how much smaller it got.
Seems less informative than growth delay.
It generally is, yeah. Because shrinkage can happen for lots of reasons besides just cell kill. Inflammation could go down, dead cells get resorbed, fluid shifts like edema.
Okay. Confounding factors.
Exactly. So while it shows something is happening, it's less specific about the actual anti-tumor effect compared to growth delay or the survival assays. But it's easy to do, so sometimes it's used.
Okay. So we've covered a lot of ground here with these in vivo assays. Stepping back, what's the big picture? Why are these assays so important?
They're absolutely fundamental. They're the crucial bridge, really, between basic lab science, the in vitro stuff, and applying things in human clinical trials.
The translational pipeline.
Exactly. They give us that essential preclinical data. Does this new drug, this new radiation schedule, this combination, does it actually work in a living system? Does it control growth? Does it kill cells?
Right.
And like we said, growth delay especially seems to track pretty well with potential clinical benefit.
And they also help us understand the basic radiobiology, but in that more complex setting.
Or sure. We can study the 5 Rs: repair, repopulation, reoxygenation, redistribution, radiosensitivity using these models. TCD50 reflects the net result of all those things needed for cure. Growth delay shows the balance between cell killing and repopulation. You can even design experiments like split-dose studies using these endpoints to quantify repair happening in vivo.
Really powerful for investigating mechanisms, too.
Yeah.
What about the cancer-stem cell connection?
Well, the thinking is because TCD50 requires permanent tumor eradication. It's probably a good measure of whether your treatment is killing those really resistant cancer stem cells. If the tumor doesn't come back, you likely got them.
Whereas growth delay might be more about the bulk tumor response.
Potentially, yeah. Growth delay reflects the overall slowing, which might be dominated by killing the more sensitive bulk cells, though stem cells definitely play a role in regrowth, too. TCD50 is perhaps a tougher, more stringent test of hitting that resistant population.
Okay, that makes sense. But we always have to remember the limitations, right? Mice aren't humans.
Absolutely. Critical point. Animal models are not perfect replicas. The microenvironment can differ. The immune system interaction is often different or absent in xenografts.
Right.
Tumor biology itself might differ between species. So while these assays give us invaluable data, you always have to be cautious translating directly to people.
Interpret with care.
Yeah.
But still, indispensable tools.
Couldn't do without them for developing new therapies.
Okay, fantastic overview. Let's crystallize some key takeaways with our clinical pearls. Sounds good. Pearl number 1, TCD50. It's the dose for 50% tumor control in preclinical models, and it likely reflects killing all clonogenic cells, including cancer stem cells, in those controlled tumors.
Got it. Pearl number 2.
The TD50 assay and calculating surviving fraction from it, that's our in vivo equivalent of the lab clonogenic assay. It quantifies cell kill within the living organism.
Excellent. And Pearl 3.
Tumor growth delay. Widely considered the best preclinical predictor of therapy effectiveness, often correlating well with clinical remission rates.
Perfect. TCD50, TD50SF, and growth delay. Key concepts. All right, ready for a board blitz?
Let's do it. Question 1. Researcher irradiates mouse tumors. Control TD50 is 10 cells. After 20 GJ, TD50 is 100 cells. After 30 GJ, TD50 is 1,000 cells. What's the surviving fraction after 30 GJ? A, 0.1, B, 0.03, C, 0.01, D, 0.001.
Okay, surviving fraction is control TD50 over irradiated TD50. So 10 divided by 1,000, that's 0.01. Answer C. Correct. Question 2. Which assay involves IV injection of tumor cells into recipients and counting lung colonies? ATD50, dilution assay. BTCD50 assay, C. In vitro assay, D, lung colony assay.
IV injection, lung colonies. That screams D, lung colony assay.
You got it. Question 3. What's the term for the time difference for treated versus control tumors to reach a specific size? A, tumor regression time, BTCD50. C, plating efficiency delay. D, tumor growth delay.
The difference in time to reach a size.
Right.
That's the definition of D, tumor growth delay.
Nailed it. Final one, question 4. The radiation dose needed for 50% local tumor control in a preclinical model is called ATD50, BD0, CSF2, DTCD50.
50% control, that's going to be DTCD50.
Excellent. 4 for 4. So, quick summary. We hit the major in vivo assays. TCD50 for cure control.
Right.
TD50 and lung colony for cell survival and metastatic potential in vivo. In vitro for that hybrid survival measurement.
Yep.
And tumor growth delay as that key predictor of treatment effectiveness.
These really are the tools that bridge the gap from the lab bench to the clinic, helping us understand how treatments work in a living system and picking potential winners.
Absolutely. And understanding them helps make sense of preclinical data and why certain treatment approaches are developed. TCD50 connecting to stem cells, growth delay to clinical response, really important links.
Couldn't agree more. That was a fantastic deep dive. Thanks so much for breaking all that down.
My pleasure. Always good to revisit these fundamentals.
For everyone listening, you can get more practice, including oral boards prep, over at radonsmartlearn.com. And Definitely subscribe to Rad Onc Smart Review so you don't miss our next deep dive. Thanks for tuning in.